Hello, in the following video we will see the factors affecting rate of photosynthesis, and these are the questions given in the exams, so we will be solving them and hopefully it will be very clear to you, right? So let's start. The question says the rate of photosynthesis is affected by several environmental factors. Maybe you can just name them so we be at a better place, right? So let's just name them the factors that affect photosynthesis. Okay, factors affecting photosynthesis. Factors affecting photosynthesis. One, amount of light. 2, temperature. 3, carbon dioxide. Amount of carbon dioxide, right? Then other ones, amount of water, maybe amount of water present, amount of water, etc. Okay, these are some of the factors affecting photosynthesis. Now in this case we are supposed to explain why the rate of photosynthesis levels out at 30 degrees. So as you can see, it goes all the way here up to when it goes at 30 degrees, it levels out, it becomes constant. Okay, so why does it become so? So probably up to 30 degrees the rate of photosynthesis depended on temperature. Okay, the limiting factor was temperature. Now if you are not familiar with this word, maybe I can describe it to you. Limiting factor. Okay, this simply means a factor without which this temperature, this reaction, limiting factor means it's the factor which affects the rate of photosynthesis. If this is not there, then the rate will not go forward. Okay, and that's what happens here. Since temperature is limiting till 30, and then it is no longer limiting after 30, so the rate of photosynthesis becomes constant. Okay, and also this it might be because there are some other factors are limiting after 30 degrees, like carbon dioxide. Say there is no enough carbon dioxide anymore now, so even if you increase temperature, the rate of photosynthesis will not increase because the carbon dioxide is already limiting, is limiting as well here. Okay, so I hope the concept of limiting is clear. It means it is sort of blocking the, without this the reaction, the rate will not go forward, will not increase up to the time when this substance is present. So it's like affecting. Okay, then so the answer of this question will be either carbon dioxide or light is limiting, or you can say the temperature is no longer limiting over here. Then we have the next question says, on figure 8.1, continue the curve to indicate what would happen to the rate of photosynthesis if the temperature was increased to 70 degrees. So we know that in photosynthesis there are enzymes, right? You're familiar with that. There are enzymes. What are examples of enzyme? We can mention, if you remember, we have Rubisco, which is there in, this Rubisco is there in Kelvin cycle. Okay, Rubisco is there in Kelvin cycle. So this Rubisco will sort of be, after, if we increase this temperature to 70 degrees, maybe you can use another color so you don't get, if we increase to 70, there will come a point, not this way actually, this will go like this first, it should go straight, okay, but then there will come a temperature where this Rubisco will become denatured. Okay, as we know, the enzymes become denatured. So if enzyme become denatured, means what? Means its active site no longer is able to bond or form a substrate, active enzyme substrate complexes. Therefore, this even if you increase temperature, since the enzyme is already denatured, is starting to get denatured. As you increase temperature, it continues to get denatured. So if the rate goes all the way to 0, okay, I mean it goes down. It doesn't go really down. It goes up to this point, okay. Then maybe more temperature will make it go really low. So that's the thing. It lowers down when it is reaching when it is approaching 70 degrees. Okay, you should have that idea. Explain why you have continued this graph in this way. We have explained, because the enzyme gets denatured. Okay, that was the question number 1. I will see you in the next video. Thank you for listening.